Good morning, everyone. My name is Fabian Castro. I am a fourth year graduate student here at the University of Texas at Dallas for the Chemistry and Biochemistry Department, specifically speaking in the Gas and Smith lab. Um, Today, I would like to share with you some of the very cool results that we've recently published in collaboration with another lab here at UTD, where we focused on the stabilization of lipid bilayer assemblies through the immobilization in a crystalline exoskeleton. Okay, but before we jump into topic, I would like to give you a brief introduction about proteinaceous materials. As we all know, proteinaceous materials or proteins are extremely important for all sorts of biological processes. However, these proteinaceous materials have the disadvantage that they are only marginally stable at physiological conditions. Um, that means that whenever you take them out of their comfort zone, they really suffer. They really encounter a lot of issues, and those issues result most likely in the alteration, denaturation, or misfolding of their uh, structure. A lot of work has done, well, a lot of research has been um, focused on trying to find better ways to stabilize these proteinaceous materials against these conditions that tend to affect their structure. However, um, there's still a long way to go in this particular area of investigation. We know that most of this particular proteinaceous materials require, again, a very specific environment to be happy in. So our work has attempted, or the main focus of our work was to find ways to provide the means for stabilization for this proteinaceous materials in a very cheap way, in a simple fashion that, again, would allow us to preserve the stability of this proteinaceous materials without really altering their structure. So to that end, we turn to metal organic frameworks. For those of you that are not familiar with the metal organic framework, a metal organic framework is basically a coordination polymer that is constructed from the interaction of an organic linker and a metal ion. Okay, metal organic frameworks come in very different flavors. You can tune their properties. You can synthesize many different sources of metal organic frameworks by virtue of changing the metal center and the organic link. Throughout my talk, I will be specifically talking about a subclass of metal organic framework, which is known as Zeolitic Imidazole Framework 8. What are metal organic frameworks used for? Well, in the beginning of the metal organic framework era, this particular materials were used for industrial applications such as gas storage, separation, energy storage, catalysis. More recently, though, these materials have been implemented or used for more biologically relevant applications, as we see from the slide, for biological imaging, and more importantly, and related to my talk today, they have been used for this so-called biomimetic mineralization process. Okay, again, specifically talking about CIF8 or Zeolitic Imidazole Framework 8. This is a coordination polymer constructed from the tetrahedral interaction between zinc and 2-methylimidazole. It is a porous material with flexible synthetic conditions. It's highly stable. Um, and very interestingly, a lot of research has used Zeolitic Imidazole Framework 8 to stabilize a wide variety of proteinaceous materials. Okay, and the way that the synthetic conditions or the way that the reaction is carried out is quite simple. Again, we have 2-methylimidazole and zinc acetate. As I was saying earlier, the synthetic conditions are quite flexible. That means that the synthetic conditions can be carried out at room temperature using water as a solvent. It doesn't get any more protein friendly than that. And what happens is that this proteinaceous material that you put in solution interacts, the negative surface of the protonation material tends to interact with the zinc ion, promoting nucleation or promoting the moth to form around it. Again, the protonation material. In result, the protonation material ends up encapsulated within ciphate, and as a function of that, its mechanical thermal properties are significantly enhanced. We've seen, again, in the past 5, 6, 7 years, a lot of people have used this ciphate strategy to provide protection to sensitive materials such as viruses, therapeutics, whole cells, and even nucleic acids. Okay. In our case, our work was, again, a collaboration with Dr. Gabriel Maloney here at UTD. He's a transmembrane protein expert. He has a lot of interest in finding ways or finding the modes of operation of this transmembrane proteins in cells. A very, very, in very broad terms, a transmembrane protein can be stabilized in so-called proteoliposomes, okay, to study its transmembrane or the translocation of the metals in and out of the cell. So a proteoliposome, again, is a liposome with membrane proteins embedded on the lipid bilayer. Why do we do that? Why would we care about doing this? Well, you have to extract the transmembrane protein from its native-like environments and still provide the means to study its translocation capabilities. So in this particular project, again, we focused on trying to study the potential protection capabilities that CIF could provide to this very sensitive systems. So the idea was very, very simple. Could we grab, or the question that we're trying to answer with this research was quite simple. Can we grab a lipid nanoparticle encapsulated in ciphate, allow it to, or protect it against environmental harsh conditions that would otherwise destroy it? Can we remove the ciphate and remain, and this nanomaterial could retain its native-like properties? Okay, so important for me to mention that this particular project was, again, a collaboration with this 3 amazing scientists that I have labeled in my slide. And more importantly, we systematically progressed throughout this study to find ways to stabilize not only lipid nanoparticles, but also transmembrane proteins and so-called proteoliposomes. So let me walk you in, or let me share with you some of the very, very cool results that we got along the lines of this project. To begin with, we started with just blank liposomes. These are basically lipid nanoparticles that are constructed by extrusion. Okay, there's a combination of 2 lipids that are used for these materials, are L-phosphatidylcholine and E. coli lipid extracts. So the idea, again, was very straightforward. Can we encapsulate these materials in CIF, stress the material, and still afford the native-like liposome? Okay, so we tested different conditions for the growth of CIF around this material, and we were very, very happy to find that we could encapsulate these materials within CIF. But not only that, after stressing, we were very happy to find that the material, whenever it was protected with the CIF coating, it could resist temperatures that would otherwise destroy it. As we see from this dynamic light scattering curve, whenever we had the CIF coating protecting the material, the size, the sparsity didn't really change. As you can see, we heated up the liposomes at 55 degrees Celsius, and in the presence of the CIF coating, the material retained its amount of sparsity, just like the pristine liposome. Whereas when we had these materials either just sitting at room temperature for 48 hours or heated at 55 degrees Celsius for 15 minutes, without the CIF coating, they greatly suffered, as we see. Their size heavily incremented as a function of lipid fusion or aggregation. We not only did dynamic light scattering characterization of these materials, we took it a step further and we looked at transmission electron microscopy. And the idea, again, is very simple. Stressing at room temperature for 48 hours at 55 degrees Celsius for 15 minutes, at 80 degrees Celsius for 5 minutes, or at room temperature for 20 days. Okay. As you see from the transmission electron micrographs, the material was preserved its structure as a function of the SIF coating, whereas the materials that had no SIF coating really suffered from this particular conditions. Okay. So the next question that we wanted to address with this project was, okay, we have shown that we can easily encapsulate these materials in the SIF coating, that they are indeed protected. However, we wanted to determine what exactly was promoting the SIF growth around these materials. So we ran seeded potentials in ICP-MS, and we came to the conclusion that the negative surface charge on the liposome and the positive charge of the zinc ion served as a nucleation point for the formation of these materials, for the formation of them off around the liposome. We also tested cationic lipids, and we came to the conclusion that the cationic lipid, or in the presence of a positive charge, this nucleation point was not as favorable. Okay. We then tested proteoliposomes again, just to emphasize. A proteoliposome is a liposome, native liposome, that has transmembrane proteins embedded on the lipid bilayer. Why do people even care about that, or why would you want to do this sort of article? Is, well, very simple, to learn how transmembrane proteins operate in actual cells. In our case, we use two different protein models, transmembrane protein models, a copper transporter and an iron transporter. And the experimental design was extremely straightforward, as we did with the blank liposomes. The idea was just to grab this proteoliposomes, encapsulate them, stress them, and then exfoliate them. After exfoliation, the idea was to study their structure and their transmembrane protein capabilities to see whether or not the SIF had indeed protected them against this environmental harsh conditions. So again, we tested 25 degree room temperature for 48 hours, 55 degrees Celsius for 15 minutes, and 80 degrees for 5 minutes. CUP-A is a copper transporter, and we were able to, after exfoliation, quantify its copper transporting capabilities. And as you see from the lower panel on the left, in the presence of SIF, CUP-A was able to resist this heating process, whereas without the SIF, really, CUP-A really struggled. It basically denatured. We have in the middle panel this dynamic light scattering data of IRT. IRT is a iron transporter. So our characterization reveals that, again, in the presence of the SIF coating, this proteoliposome retained its size. And finally, we see this McCabe-Menton graph of CUP-A, showing the two different synthetic conditions that we use throughout our experiments. And again, showing that in the presence of SIF, CUP-A was able to still translocate copper, whereas in the absence of SIF, the native CUP-A, after stressing it, really had a hard time, or basically could not function as well. We took this experiment into a completely new level, given the conditions that were taking place with COVID. We wanted to study, well, can we use these materials or stress them in conditions that would resemble what a vaccine, what a protein therapeutic would go through after its manufacturing process. So we encapsulated these materials, proteoliposomes. We sent them off from Texas to Rhode Island. They came back and spent approximately 3 months sitting at room temperature in the lab. When they came back, we exfoliated the SIF coating to recover the proteoliposomes, and we were very, very happy to find by transmission electron microscopy that these materials, when compared to their pristine counterpart, retain their size. Whereas the materials that were shipped that had no SIF coating, again, showed membrane through fusion aggregation. But even more excitingly, we were able to quantify the transmembrane protein capabilities using, again, COP-A, the copper transporter. And again, we were very, very excited to find that this material, the SIF coating, could really protect the transmembrane protein and the proteoliposome. It allowed them to retain their structural integrity even after so much stress. So that was quite exciting for us. And well, talking a little bit about this global pandemic that we've gone through, we have come to find out that yes, lipid-based vaccines are amazing, that they have high loading capabilities, that they're biocompatible, biodegradable, and they're quite, well, they're somewhat easy to make. However, again, these are very susceptible to their environment. They require this extensive cold chain process that basically guarantees refrigeration, low temperature refrigeration throughout their transport, storage, and handling before they are injected into a patient. So that can really, if we pay attention or we take a minute to analyze this cold chain process, well, that can really affect the distribution of vaccines in a, for example, a third world country or places where electricity is not necessarily guaranteed. So again, with this particular project, we were very excited to be able to provide the means or a experimental flow that could eventually be used for the for the encapsulation of lipid-based nanoparticles, such as vaccines, that could eventually not require refrigeration and could be distributed in places where there's no electricity, or again, electricity is not necessarily guaranteed. So with that, I would like to conclude by just, again, telling you that we have developed an affordable, scalable method for enhancement of thermal and mechanical stability of liposomes, transmembrane proteins, and proteoliposomes, and to share with you that we are currently trying to take this experimental design, take this idea, and implement it using lipid-based nanoparticles, more specifically the Moderna lipid nanoparticles that are in the market. I would like to thank my advisor and all the people that help out throughout this long journey with this project. Again, thank Dr. Gabriel Maloney and his great team, the funding agencies that make this possible. And with that, I would like to open the floor to answer any possible questions you may have.
All right. Thank you to Fabian. I think Fabian can't hear me still, so I'm going to ask the questions via chat. So for the audience, be patient, and Fabian, I'm sure, will be patient as well. So the first question is, what do you attribute the stability of the ZIF structure to?
Okay, first question. What do you attribute the stability of the SIF structure to? Okay, so we look at, to answer your question, SIF itself, again, as a coordination polymer, allows this, or it's formed, it forms this coordination bonds between the zinc and the methylamidazole And this stability comes from such a strong interaction between the two. Um, I don't know if that kind of answers your question, or you could have a follow-up question. Okay, is there a particular advantage to using CIF8 versus other MOFs? That's a very good question. Okay, so there's hundreds of different MOFs that are out there used for industrial applications. Some other MOFs that are used for more biologically relevant applications. We have, for example, New 1000, which is a zirconium-based MOF that Dr. Farha has used for protecting therapeutic models such as insulin against harsh environmental stressors. However, the advantage of using CIF versus other MOFs is the fact that its synthetic conditions are very mild. You can synthesize CIF8 or encapsulate, well, basically afford CIF8 formation in water at room temperature. Whereas other MOFs, like the one that I was telling you about, New 1000, requires very, very harsh synthetic conditions. In some of the cases, they use modulators that are extremely acidic. So if we think about it, this strategy to use other MOFs is somewhat um, hindered by their synthetic conditions, whereas CIF itself can be very, very easily produced at room temperature with protein-friendly solvents. That's an excellent question. Are there any other concerns with viability of moss or other things? Okay, so we're actively investigating the usage of CIF itself for more in vivo applications, and we've come to the conclusion that CIF itself can be safely injected in vivo uh, at a very specific concentrations. We normally use approximately 10 to 20 micrograms of CIF per injection, and this has shown through cytotoxicity assays that is not toxic. And we've also shown with in vivo models that the body can really handle these materials. So in the case of CIF, we're good, but with other MOFs, yeah, it can be a very different story. Thank you so much. Have a wonderful day.